No, it's perfect, it's perfect, it's perfect. No, it's perfect, it's perfect, it's perfect.
Want me to hit play?
No, I'm already recording, I'm already recording.
I
don't either, actually. I see him like, what's up?
No, but we were friends before. But I never see him anymore.
Hey.
Stop.
Don't forget it.
So what I do is listen to that speak and search.
Yeah. Okay. So yeah, nutshell, all of you know about it. Thanks so much for coming out. So we were going for 50%. I don't think we quite made it, but still this should be a good presentation. Hopefully you guys can give some feedback and we'll reevaluate as time goes by. And I'm also very apologetic to all of you that came out at 8. I'm so sorry. There was confusion with the time and I'm so sorry for that. But we're all here. Hopefully we can enjoy the presentation. We can see how this goes. So yeah, again, I'm a junior in college. Everything that you're going to learn may be bogus. None of it may be true. We don't know. But, so just disclaimer for, accuracy reasons, which we have to give out. The next thing is I've used some pictures from this textbook. It's a very good textbook in my opinion. But I won't be going through the mundane details of each, genetic of each process because I think that's kind of boring. And this presentation will be more examples in real life than anything else. So I think you guys will gain a lot from this. I got really excited just making the presentation, so hopefully you guys like it. Also, all of you are a little shy group. I think this club as a whole tends to be very shy. So just speak up. If you guys have something to offer, just feel free. No need to raise your hand. Just offer your opinion. If you have a good example, offer it. If I make a mistake, call me out. Yeah, so the main goal is to expose you to how diverse genetics is, because there's so much about us that you can explain from genetics, and also a lot that you can't explain. But alas, let's begin. So what is genetics? Study of inheritance. What's really interesting is that genetics across the board is very similar. So 98% of all organisms that have ever lived are extinct. So genetics forms a evolutionary success record of sorts. So every organism that couldn't survive with its own genes was weeded out and passed away. So all the survivors lived on. And all of us in this room are the products of the survivors millions and millions of years ago. So I think that's very interesting given the fact that a lot of organisms have died. So any animal that goes extinct just was not fit for the environment. And we can blame their genes and their genetics. Also, we are only 1.5% different between the chimpanzee and us, and humans. So that's a very tiny percentage. But let's continue. So this won't be a presentation full of text. This is just in the beginning, just to situate ourselves. So essentially, there's this big study done of one guy who's very introverted, and there was another guy who's very extroverted. And what happened was radically, out of nowhere, they had a huge change in their personality. And this was due to one single mutation in one gene, a serotonin gene, serotonin receptor. That's a big hormone in our body. And so this small SNP, SNP, which I'm sure most of you have learned by now, was a one base pair mutation in their genome, which caused such a radical shift in their personalities. And this is an actual article, which I can give you guys later, but just a brief summary. Going on, how does genetics influence our life? So, cleft chin, genetics. How your thumb bends. Can anyone do this, by the way? Yeah?
Not really.
Yeah. Widow's peak. Does anyone have a widow's peak? No? Okay. Can anyone do this? Who cannot do it? Try it now. And I think, I believe I was waiting for eye color here. Yeah. Also, earlobe. Look at your own earlobes. Do they vary from the person next to you? Dimple. Do you have a dimple? Yeah? That's genetic. Do you? No? A father and his son, when they place their hand down, their pinky stays up. Does your pinky stay up? Just test it now. What do you guys see? Yeah? Clasp your hands without thinking about it. Which hand's on top? My left hand is on top, my left thumb. Does anyone have a right thumb on top? Yeah? Okay. Precisely. Outliers. Cool. Going on. Back to the effect and influence of genetics. So it impacts literally everything about us. So down to our religiousness, down to our sexuality, how driven we are in those regards. There's Asperger's syndrome. So Asperger's is a very stringent focus on one subject coupled with difficult social interactions. So does anyone know anyone with Asperger's? Have you ever noticed or witnessed someone with this syndrome? What do they act like? Like they look away from you. They tend to be very smart, focused on one subject. Coincidentally, so.
Certain colors bother them, like color yellow.
Really?
Yeah.
Okay. Another thing is they're usually not religious. They're all atheists. Why? Who knows? But there's a link between the mutations that cause Asperger's and piousness, how religious we are. And this is in literature, so I'm not just making this up. So the new trend in genetics has been that it's not just your genetics that influences who you are and who you can become, but also your environment, where you grew up, how caring your parents were. And so let's just see how that came to be. So we can think about John Watson. So just take a second to read this really quickly. Essentially, he's saying that regardless of your genetics, your environment determines who you become. So you can, from a genetic point of view, you could be a failure, but given the correct environment, you can thrive, right? So think about, we'll get to this example in a second, but just think about this. So there's an extreme opposite end, where your worth is your genetic worth. So this guy's Francis Galton, he created eugenics and genetic determinism. So selective breeding, right? So what that means is, read up here. Unfit human traits are inherited in the same way as color and guinea pigs. If all marriages were eugenic, we could breed out all this unfitness in 3 generations. So he's saying we can eliminate all of our bad qualities in 3 generations if we selectively breed, right? This is what we do in dogs and in lab rats and such. Yeah, so here's a little scale. So great genetic worth and social worth, you're skilled, you're intelligent. And on the opposite spectrum, you have no genetic worth and you deserve to be weeded out of society. So let me ask you guys, do you think it's possible for someone with low social and genetic worth to be successful? Can an alcoholic become successful in our world? What do you guys think?
Yes.
Say something. Yes? Okay. So I was hoping you guys would say no, but there's a very, very interesting example. Hopefully you know who this is. George Bush. He is an alcoholic. He has alcoholism in his genes, but given his environment, he became president. And so if you look up Bush alcoholism, you get a ton of results. So he's a heavy drinker. Dad was an alcoholic, but he still became president given his amazing environment, right? Going on, so I've talked about influence of genetics, how it impacts us. What do you think a gene actually is? And so I don't have anything else after this in terms of words, so why don't all of you answer? Margot, any ideas?
Something that codes for a specific trait.
Okay, that's good. Any other? It doesn't have to be so scientific, it can be anything. DNA, okay. So let's move on. Can you repeat your definition?
Something that codes for a specific trait.
Yeah, so something that codes for a specific trait. So what we find is that groups of these little coding segments form one property or structure, right? So height is a very good example. So going back to the genetics plus environment, our physical appearance or trait is a combination of our genetics and our environment. So we can talk about our genetics, the height of our parents. Are your parents tall? Are they short? Then look at yourself. Are you tall? Are you short? Monsi, are your parents tall or are they short?
They're short.
Okay. Who has tall parents but is short now? Or the opposite? Who has short parents but is tall? Yeah? Okay. So that's genetics. You probably, there's a high chance that you should be short, right?
Yeah.
So think about your environment. What do you eat? Do you eat high protein? Yeah. Okay, so you have a good environment, you're well fed, you don't suffer from malnutrition. What we find is that people in Asia have very bad nutrition, and so they're short. But when they come to the US, they're taller, you know, like have more muscle mass and such. Here's another interesting question. When do you think the environment begins? Is it only dependent upon what you eat, or is there something beyond that? For example, did your own environment start when you were born, when you were conceived, or even before that? Before?
Because, like, studies have shown, like, in this one village, like, people whose grandparents starved or didn't starve at a certain time, like, it affected, like, their grandchildren's tendency towards, like, being over or underweight.
Yeah. She's talking about the Dutch winter, right? Emily?
Yeah.
Was it the Dutch winter? Yeah. So what happened was in World War II, a village in Holland was captured, and they were starved for months. And what happened was that the women that were pregnant in this village, they had babies that had a thrift gene. So these babies were very adept at storing fat. And you know, with that comes a lot of health problems. So obesity, heart attack, type 2 diabetes, and so forth. But all other babies who were born afterwards had no such problems. So because the mother was starving, her baby had a tendency to, you know, take what it could get, right, and store it. So we find that the environment begins before our birth, and perhaps before our conception. So how do we study genetics? How can we model our own genetics in animals? How can I use a rat to see the effect of a drug on my own depression? Yeah. So what we find is that there's a lot of traits are conserved, a lot of genes are conserved throughout history because there is one primary ancestor. Are you guys familiar with this? Yeah. So if you're not into evolution, you probably don't subscribe to this, but I'm assuming all of you do because you're into science. So yeah, so nature is not creative. That's a big thing. So going on. Rats. We induce a mutation in a rat that is present in humans. Look at the spot on the top. Spot on the top, right? What's going on here? Hair, I think? This is more hair. Look at the stomach. Heart-shaped pigmentation on the rat and the stomach. We can also look at the forehead. So we can use mice because they're 80% identical to us. And going back to how I can use a rat to model depression in myself. What do you guys think? How can we use rats to model depression? Can we ask rats how they're feeling? No? Do you guys have any idea how we...
But like they exhibit kind of behaviors that we can interpret as depression. Like when you throw one rat that has been like put through stressors that induce depression, they like tend to exhibit passive behaviors like floating. Whereas if you throw a rat that isn't depressed into a bucket of water, they struggle with it.
Yeah. Okay, Emily, you want to give this presentation? So she's talking about it's like a floating test. So you take a rat, you throw it in some water, you see how long it takes for the rat to just give up. And that's similar to the despair index. You take the rat and dangle it upside down. How long does it take for the rat to just stop struggling? Because the rat will try to climb back up, climb back up its tail. But if they're depressed, they'll give up sooner. So what you do is you give this rat an antidepressant. If it works, it'll struggle for longer. So it's kind of a morbid test, but that's how we can model unusual behaviors in humans and other animals. Going along that, how do we measure speech impairment in other animals? So what we find is that the African gray parrot has the same gene as us, called the FOXP2 gene. So what do parrots do that humans do?
Speak.
They speak, yeah. So because this parrot has the FOXP2 gene, it can mimic us, it can talk. Humans, if you break the FOXP2 gene, or if you inhibit it somehow, we have speech impairment issues. Make sense? So this is convergent evolution, you see? So different traits being, traits arising despite different evolutionary paths. So just a brief thing about Mendel. Have you all heard about Mendel? Who has not heard about Mendel? Okay, good. So I won't get into this much. But essentially, cross P's learn about the basics of inheritance. So you can apply that to everything: dogs, vegetables. You apply it to a pepper, you get red, brown, yellow, green peppers. Have we seen brown peppers before? Do you see brown peppers at the supermarket? Why not? Is this wrong?
No one's gonna buy brown pepper.
Yeah, it looks gross, right? It's negative selection. No one, sorry, it's artificial selection. No one wants a nasty brown pepper, unless for the novelty. So anyway, takeaways, what's an allele? It's a variation of a gene. What's a phenotype? The physical appearance. Typically, the phenotype or the physical appearance is based on the genotype, the genetic makeup, which is based on the alleles, which two alleles from each parent came together to form a trait. So homozygous, heterozygous, you typically, you have identical alleles of the same gene. Heterozygous, non-identical. Is anyone unfamiliar with this? Cool, so this is not groundbreaking. What is groundbreaking is that you can have different genetic makeups but have the same physical appearance. And that's a big deal because it makes it hard to determine what genes cause what. But we can make up for that. So now let's just have a small shift into reproduction. So asexual versus sexual, what do you guys think? Which is better? Which is more advantageous from an evolutionary perspective? Which one do you think would be easier? So, and go ahead.
Well, asexual is good because it's the advantage. It's like you need a male and like asexual, so they can produce twice as much as sexual organisms. And if you like put one asexual individual into a population of like 100 sexual reproducing, they can like basically overtake the entire population in 7 generations.
Okay, so would you say that's a pro or a con?
From an evolutionary standpoint, probably.
So it's very easy to reproduce. So, any other hands?
Wouldn't sexual reproduction be better for evolution since there's genetic recombination?
Yeah, so there's more diversity in sexual reproduction, yeah. But the rebuttal is, how much energy do you expend for this diversity? Is it worth it? All this energy you're spending in? So if all of you are familiar with meiosis, meiosis, it's a long process full of like recombining, and we'll get into more complex stuff later, but is it worth it? So we find it is worth it. It is worth extra energy. But what's interesting is that there are other methods of sexual determination, so that weren't asexual, that weren't sexual, but they have to do with the environment, right? So alligators, how is the sex of an alligator determined, or a crocodile? The position in the stack. So if it's on the periphery, it'll be colder. And if it's colder, there'll be this. This is actually for a toad, which is the opposite. So let's just go for the high temperature. So if it's colder, it will lead to high aromatase activity. It'll become a female, right? If it's on the inside, it'll be hotter. It'll become a male. So this was advantageous for crocodiles. For toads, it's the opposite. This is just some, DMRT1 is the gene that is expressed or is not expressed. And so this, I'm not getting into biochem of this, but the higher the temperature, the higher the proportion of males for alligators and crocodiles. It's the opposite for toads. Or sorry, turtles. And so here's a nice little easy graph. Yeah, so, can we apply this temperature thing to all environments? No. So the sea is typically the same temperature in one area. And if marine animals reproduce this way, they wouldn't have a lot of diversity in terms of sex. It will be all males and all females. And so over time, evolution has decided that sexual determination is best for diversity. And that's one of the reasons that sex is so common in a variety of species. We'll have an example later on. Any ideas what this is? Yeah. So going on the topic of sex, this is a nice little picture of sperm entering the egg. What's interesting is as soon as one sperm enters, this zona pellucida forms over the egg, which prevents other sperm from entering. This is just a really cool picture that I think you should set as your background. Drosophila, sexual determination. I may get into this later, I forgot, but Drosophila have a very intricate dance. So that's a fruit fly. The male has a very specific routine and a ritual for the female. And what you find is that there are more proteins in the male for this dance than the female because he's doing all the work. The girl's being wooed in a sense. And so this is just a funny picture of how they actually made this is a real deal. This is a real thing. Just thought you should know. Yeah, so back to sex. We have 23 chromosomes in humans, or sorry, 23 pairs of chromosomes for 46 chromosomes. And we can model these in a karyotype, so just a picture of all these. And we can detect small inconsistencies and problems. So, for example, Turner syndrome leads to this. So what do you guys observe? You can look at this to see what's what's going on. So poor breast development, apparent here, apparent there. So, and that's when you have X0. So, or sorry, you have 2X0. Yeah? 45 that. 45? Okay. Or so you have two X chromosomes and not so much and Y, XXY. That's Turner's.
I think that's fine.
Oh, okay. Sorry, messed up the pictures. Yeah. So thank you, Victor. But going along that, so if we just look at the Y, we notice that it's smaller, right? So the X and Y, females XX, males XY. Is this new to anyone? Cool. We find that the Y chromosome is smaller because, to be honest, it has less information than the X chromosome. But the most pivotal region on the Y chromosome are on the ends. That's the SRY region. So the SRY has a big role in everything. So down to cruelty, aggression, criminality. If you have more SRY expression in your body, if SRY expresses more proteins in your body, you're more likely to be a criminal. You're going to be more aggressive. So part of the reason why guys are more aggressive, if females are not. So yeah, just difference between the two. So now let's get into interesting stuff. So in genetics, we have a cascade. Have you guys heard of an enzymatic cascade? So one thing leads to a few other things, which lead to a few other things. In genetics, we have master control genes, which control the transcription or the expression of everything after them. So I'm not really going to get into this because this is central dogma. You guys have hopefully learned this. If not, I can spend a small amount of time. No? Okay. But master control genes. Think, what happens if we break something over here? Do you think there will be a big effect? Yes, no, maybe so?
No.
No. Perhaps. What happens if we break something here? Big effect?
Yeah.
For sure, right? Because all these other things are knocked out, aka are not working. So MyoD is a very good example. MyoD is responsible for muscle cell synthesis. And so, basically, you have MyoD, the master control gene. It is inhibited by myostatin. So when myostatin binds to a myostatin receptor, MyoD shuts off temporarily. So why would we want to stop muscle production? What do you think?
Starvation.
Yeah, starvation. You can think of anything. Stress is something you can blame for anything at all. But yeah, so we have a cascade. So what if MyoD breaks? What do you think would happen? If our master control gene for muscle is no longer working, we will no longer make muscle, correct? Yeah?
You break all your muscle.
You won't break your muscle, you're going to stop producing muscle. And I get if MyoD has a role in maintaining muscle, then yeah, you'll just waste away. So think about the opposite view. What if myostatin breaks? The inhibitor of MyoD. If myostatin is broken, MyoD is constantly working. So what do you think would happen if you're constantly... Yeah, you're constantly making new muscle. And there is a disease where this happens. Bam. If myostatin breaks, you have a swole ass kid, okay? His name is Liam Hoekstra. He is 8 years old in this photo. This picture was taken 3 years ago. Like, he has not worked out a day in his life. It just happens naturally. And so there are repercussions, you know, like, if you're using too much muscle, you need to eat enough so that you have enough calories for everything else. You can't just be making muscle all the time, right? So, just a really interesting case study. Liam Hoekstra, if you want to look him up. So,
How come it's only like, how come it didn't like build muscle in his face or anything? Like, it's just concentrated in his arms and chest and stuff.
yeah, so that's a good question. But I would say that, you know, you have larger muscles everywhere else. Like, I imagine his legs will be massive. So, yeah, that's a good question. I'm not too sure. Does anyone have any ideas?
Like he eats.
Like,
right?
you can get like a meaty face. Like,
Yeah.
if you see boxers, they have like, you know, monkey ape-looking faces. But you can blame delayed evolution in a sense. I don't have a slide on that, but if we continue this, I will. I don't really know, but that's a good question. So yeah, let's look into that. So yeah, this MyoD, again, you know, genetics, genes are conserved, and there's convergent evolution in a lot of animals. So we can put this in cows. We get a very, very large cow. So this, they actually do this in the real world for like, you know, genetically modified foods and such. You break genes so they grow bigger. Cows are one of them. Okay, what am I talking about here? Right, so dogma. Yeah, so nothing. When we replicate, we want to ensure that we are not replicating mutations because we have a lot of mutations in our bodies, right? Is that new to anyone? All of our DNA is jacked up. We've mutated like crazy, and in some cases, the mutations have, you know, been in our benefit. And at the end of replication, we shorten our telomeres at the end. The telomere ensures that we are not replicating the same cell or same gene multiple times. Protein. Make sense? Yeah? Yeah? So, what if our telomeres are constantly being eroded too quickly? We have a progeria, advanced aging. So this guy's 26 years old in this photo. I'm 20 right now. I do not want to look like 6 years, right? So luckily I don't have progeria. So premature aging, you have short telomeres that only go out a few times when going culture. So your telomeres are growing very slowly and are being chopped away quicker. So what do you think would happen if we have a disease that increases our telomeres?
You would go.
Yeah, immortality. So that's what they're looking into. How can we increase telomeres for a long time? But there is an animal in nature that constantly grows its telomeres over time, and it lives for over 100 years. It's called the fulmar spectral. So this lives for over 100 years, and from an evolutionary point of view, is this a good thing for the bird to live over 100 years? Why? Yeah, that's a good, yeah. So you have more time to mate, but there's also the question, would you want your grandmother's mating? They are essentially your competition in this world. So what's your rebuttal for that?
If they're like really slow to mature, then, and if they're not like eternally living forever, so they die off before they really.
Yeah. So they only mate like once or twice in over 100 years. So it's called fulmar spectral, F-U-L-M-A-R. It's really cool, aka fulmarine. But long-lived bird mates once or twice, and because it mates so infrequently, it has to live for longer in order to succeed. That's how it was selected. It couldn't mate, so it couldn't mate in a lot of time. So the petrels that had the ability to increase their telomeres lived longer and survived. So going back to aging, we're not supposed to age in the environment. Technically, we're supposed to be killed off by natural selection. So that's why there's a lot of aging is so weird and unpredictable, because it's not supposed to happen. We're artificially increasing our lives. So here, typically in nature, we will be gone. But since we can live longer, we actually experience aging. Yeah, that's an interesting thing which I didn't think of. This is a cicada. Who is from DC, Maryland, Virginia area? Really? You're from North Carolina?
No, Virginia.
No way. She's actually in my like peer advisee group, so I've known her since I got here. Yeah. So this cicada, how long does it live? Really? 7 years? No. It lives for 14 days or less, and it takes 17 years to grow or to hatch. So the cicadas, they mate every 17 years. They pop out of the ground, mate, or are eaten because they're a delicacy. Then they burrow holes into the ground for another 17 years. They're actually expected back in the next 5-6 years, I think.
There's different species. Some are 7, some are 17, some
Really?
are 25.
They have overlapping timelines.
Oh, okay. So I didn't know that. I thought it was just 17, but still,
Those
that's
are the most annoying ones.
yeah. Really?
Those about the big ones.
They actually came out when I was 9 here, so there's like this massive craze, like all the kids in elementary school just kill them out of nowhere. But they're actually a delicacy, so you can buy them to eat for a lot of money. And so during that time, a lot of people just catching them to preserve them for culinary adventures.
Don't they come out like every year?
It's like
There's
apparently.
there's like a smaller, less common species, I guess, that comes out.
The one I'm familiar with is the one that's in Maryland, which comes out every 17 years.
Yeah, I feel like I see them every year.
Really? Where are you from?
From PA.
Oh, Pennsylvania.
Yeah.
I was like,
I was like, huh?
Alright, so going back to DNA, we find that DNA is hella stable, right? So we can actually extract DNA from Neanderthals that are over 30,000 years old. How do you think we know how dinosaurs lived? We can extract DNA from their bones. Have you guys seen Jurassic Park? Do you remember how they know? So the theory in Jurassic Park is that if a mosquito bit a dinosaur right before dying, right before it was like immortalized in tree sap, you could extract the blood from the mosquito and regrow dinosaurs. You can bring them back from the dead. So the thing is that can't happen because the DNA would actually disintegrate over time, but in tree sap at least, in mosquitoes, so the mosquito DNA and the dinosaur DNA would like mix together. You can have like a dino mosquito. So that can't work. So that's why Jurassic Park is not possible. But they have been able to extract dino DNA. I took a class on dinosaurs my freshman year. It's the only class that I think was, it's been the best class I've ever taken. The Smithsonian, the dinosaur section, was designed by my professor. Jurassic Park, the movie, my professor was the advisor. He wrote a huge manuscript for the directors so that everything was historically accurate, or scientifically accurate. It wasn't. But, anyway, really cool thing. So a lot of the things we know about biophysics come from dinosaurs. So paleontology is a huge interest of mine. When I first joined this journal, I tried to submit a paleontological article to Victor. I said, please publish this. And they said, no, this is not health science. But very interesting field, which I suggest you look into. Going on. So yeah, DNA has a half-life of 521 years at this low temperature. So where would you get this temperature? It's been buried underground, special case. But so that's still a long time, 158,000, 16 million years. So, you know, going on. What am I talking about here? So structure of DNA. Should I go into this? No? Okay, cool. I don't want to. No. Okay, so we have DNA. When it's condensed, we wrap it around a histone. Yeah? Make sense? That's how we end up with this beautiful structure. Otherwise, it's like spaghetti. It's called, what's it called? Chromatin?
Yeah.
Yeah? Alright, so now some of the things that'll help you in your articles. I'll get back to more interesting real-life stories after this. But you read a lot about promoters and activators, enhancers, inhibitors. So the stretch at the top is essentially just a chain of DNA code. You have a promoter. What do you think a promoter does? Promotes gene transcription, right? So if something binds to a promoter, so what happens is you have a protein that causes folding of the, should I draw it? No. So it basically folds over and you can bind both the activators and the promoter and any other inhibitors and such that are over here in order to change transcription. So when you read something like activator discovered in Drosophila, so that's just saying we found something that increases transcription by binding to the promoter. Does that make sense? Any questions? What a promoter is? Who knows what the Tata box is?
Does that work for fruits to starting things?
Yeah. Anything else, Monty?
Initiation of transcription.
Victor, do you want to add some more to that? Oh, that's what I was going to say. I
It's
mean,
a very new concept, the Tata box. It was like 1994. I know the person who invented it.
wow, look at you, right? Awesome.
Goldberg, Michael Goldberg.
What's really interesting is there's this law called Moore's Law, which says that, so you said really recently, Moore's Law says that every 9 months, Moore's Law says that our knowledge of technology is double that what we thought, so our knowledge of DNA is increasing exponentially. Does that make sense? So in 9 months, our knowledge of the genome now will be doubled. So essentially, we can't keep up with technology, or by keeping up with technology, we're enhancing our knowledge of the genome. And so what you said is relatively new, that's ages old now. But just, yeah. There's actually something called the Oxford Nanopore. You can, what's the word? You can record your genome by like pricking your finger. So for $1,000. So it used to be that, what's the word for making your genome? Sequencing your genome, yeah, sorry, I can't speak English. So it used to be that sequencing your genome would cost upwards of $1 million. Now it's down to like 10,000. Then 1,000 with Oxford Nanopore. So we'll see how low that can get. My genetics professor actually got his own genome sequenced, and he found that he can eat as much chocolate and fatty foods as long as he wants, because the genes he has, they have been shown to not be affected by fats and sugars and such. So he can eat whatever he wants, he can smoke as much as he wants, and he'll live a very long life. So that's like, there's more about bioethics into that, which I'm not going to get into. So yeah, what's an enhancer? It increases transcription. It can be far away from a promoter. So going back here, like it will be right next door, but we can have 1,000 base pairs, upstream. Upstream means like to the left, before. Downstream means after the promoter. Okay, blah, blah, blah. Okay, cool. Now this is interesting stuff. All right. So we have, what we find is a lot of our DNA does not code for anything. It's termed junk DNA. But recently, scientists have noticed that this junk DNA is not actually junk. It's actually used for specific purposes. And so we're going to talk about regulatory sequences later, which is very interesting. But just showing you 15% is non-coding. Repetitive DNA, like why do we need DNA segments to be repeated multiple times? Yet it still makes up 14%. Yeah, cool. Okay, splicing, do we all know what splicing is? Yeah? Should I go into it? Cool. Example of splicing, we have, do we know what, introns, exons? Introns are taken out, exons are expressed. Okay, example, calcitonin gene-related peptide, made from the same segment, but they're spliced differently. Different segments are included, different segments are excluded. So calcitonin is a big hormone. What does it do? Who knows what it does? What does calcitonin do? Can you hear something about calcium?
Is it inhibiting like calcium breakdown?
Is it? Is it inhibitory or what do you guys think? I mean, I forgot. So I'm assuming it's inhibitory. Whereas CGRP, it's in the brain. Like, what the heck? Completely different function, but it's still coming from the same segments. Why double-stranded DNA? Let's not get into this. RNA can be double-stranded. Yeah. So that's a big deal going forwards. Okay. Ha ha hoo hoo. Laugh for a sec. I'll chug some water. Yeah. Does anyone have any questions? This is actually what happened. The DNA story is just two guys testing things out. And you guys know the triple helix story, right? I think I gave it last time. Yeah? Okay, so now let's focus on regulatory sequences. What do you think that means from the name? Okay.
Sequences that regulate things.
Yeah, that's basically it. Well put. Okay, wait. Okay, actually you guys saw it. So, there are three, so the most related to humans, the two species, I guess, bonobos, chimpanzees. And chimpanzees and bonobos are so different. Like, does anyone know an example of, yeah?
So like chimps are angry and stuff, but they also, like humans, have been shown to murder each other, and like actually chase each other down and like murder each other in cold blood. And like bonobos are like super loving and they just have sex all the time.
Yeah.
They're like super happy. They're like, oh, apology sex, angry sex, like all the sex because it like makes us happy and they take care of each other and stuff. So.
Yeah, that's perfect. That's perfect. Bonobos, when there's a conflict, you have sex. If you are upset, if you are happy, you have sex. You know, like you said, whatever the mood, you like copulate to, you know, lighten the tension. And what happens in bonobos, female bonobos lead the pack. In chimpanzees, you have males, you have little groups, and you have a lot of aggression. And so humans are compared to chimps the most because, again, our DNA is 98.5% similar to theirs. And so what we found is that, oh, so another thing about chimpanzees have spines on their penises. Is this new to anyone? Have I? Really?
Barbed penises.
Have I talked about that?
As an example.
Okay, well, I didn't know this. So, another thing, so chimps have very big balls. Sorry. Thanks, Alex is going to see this later. So chimps have, you know, big brains, big balls, and they have spines on their penises, right? Why do you guys think chimps would have a barbed penis? So given the fact that chimps highly aggressive and that, you know, there's competition.
They fight with them?
I mean, I don't think you're gonna like whack your.
That was a thing.
Huh?
I thought that was a thing.
Really? Okay. Perhaps. I mean, so there are two theories that I remember. One is that when you mate with a female, the barbs are, you know, cutting inside the vagina, right? So you're kind of making a little tunnel. So it'll be more painful for her to mate with some other man, some other chimp, right? So you're kind of making a tunnel. And another thing is the spines can act as scoops. You can scoop out any semen that's inside. Okay? So why would humans not need this? Typically we're monogamous. Yeah, that's one of the biggest reasons. Chimps are not monogamous. Neither are bonobos. Bonobos are also very bisexual. So yeah. So going back, this is a huge article. It was in Nature, if I remember correctly. So over time, we lost regulatory DNA that chimps have, and because of this loss, we've lost big balls or big brains. we've lost big brains, and we've lost the barbed penis, right? So, don't you love this topic? So, regulatory alterations may be okay. Blah blah blah. One deletion removes the penile spine enhancer. Okay, anatomical loss of spines, expansion of brain regions, loss of large brain, right? What do you think a barbed penis looks like? You're about to find out. Oh wait, not yet. So the way they did this was they put again a rat, you know, mammalian genetics. So again, we lost genetic material that create spines left. So boom. So that's the little rat developing in utero with spines, and then right to the brain, smaller brain. Now we move to the awesome slide. Yeah, I've talked about this. Yeah, just take it in. Yeah. So in regards to, I had a question. Can you break the penis? Yes, you can. Not humans, but I think chimps have an actual bone. So, yeah. Luckily, that's not us. Okay, so now making a small shift. Transposons. So the reason our genetic material is so dynamic is because we have segments that actually jump, right? Transposon, jumping genes. So we have two classes. One moves DNA directly. So what it does, it flanks DNA. So you have this segment. It'll look for a certain sequence on the left and on the right. And it'll bind, and it'll make this segment jump throughout the body. So there's a lot of this activity. So we have, we can also do RNA to DNA, and we can move things that way. So MRSA, what is MRSA? iHealth people, do you know? Sandra, what do you think?
I don't know what it is.
Really? Okay. So MRSA, I think it's flesh-eating bacteria. Sorry? Yeah, it is. I was going to put a picture, but I decided against it. So what happened in MRSA was that it was a human disease at first. But, we farm. MRSA has a lot of transposons, so it jumps to cows. And what do we do to the cows? We feed them antibiotics. We give them hormones. And over time, the MRSA bacteria, or virus, is it a virus? Whatever it is, has acquired these mutations, or it's adapted. And then, because MRSA still has these transposons, it's jumped back into humans. And now we suffer from it, and we can't fight it. Corn, grapes, jumping genes cause diversity. So we can see how do we get white grapes, red grapes, black grapes. So blame transposons. Yeah, we can methylate. Oh, okay, so we're near the end. So not the end yet. So epigenetics, traits inherited on top of the DNA without actually affecting the sequence in question. Agouti mice. Now what happened? So folate is a big deal in humans. If the mother doesn't eat folate, your kids have what? Neural tube defects. Yeah. So same in mice, not neural tube defects, but you affect methylation. Methylation adds methyl groups CH3 to gene segments. And so if your rat mother did not eat a lot of folate, what happens? You are not methylated. If she does eat folate, you get methylated, and you have a healthy mouse with brown fur. If your mama does not eat, thanks Alice, again. If your mother doesn't eat a lot of folate, you're not methylated. You end up fat and yellow fur, and you get diabetes. In bees, how do we get a queen bee? So she's fed royal jelly. The royal jelly has a lot of methyl groups in it, which lead to the methylation of your genes that inhibit the formation of an ovary. So you turn into a worker bee. You inactivate your inhibiting genes for the ovary, and you turn into a queen bee. So the bees that don't eat this jelly turn into worker bees because the ovary inhibiting genes are on from the get-go. Cool. So going back to it's a lot of stuff which we're not going to get into it right now because we're out of time. But yeah, so in case you guys like this series, we didn't quite get the turnout, but hopefully you guys liked it and learned something. I have this feedback form which I will pass out. But possible topics. Cloning is really awesome. How can we deliver, is it possible for us to grow a banana with a vaccine in it and give it to all the kids in the world? Yes, and it's been done. So we can talk about that. Cloning, personalized medicine. When Obama walks into a room, he has a crew wipe everything down when he walks in, wipe everything down when he walks out. Why? There's a potential to create a highly specific toxin or bioterrorism thing that can just kill him. If they have his DNA, you can theoretically create a protein or a virus that will just kill him. And that applies to all of us. So thanks so much, guys. Hopefully this is good. Brutal honesty would be much appreciated. Write as much as you want. Meaning, please write a lot. Pass this down. And just pass. Sorry. Oh yeah, no names, most wanted. Please critique the style of lecture, content, literally anything starting. Is anyone missing one?
I have one.